Hello, everyone. Welcome. Good afternoon, morning for those of you on Mountain and West Coast time. First and foremost, I want to wish you all a happy American Wetlands Month, and welcome to the SWS webinar for the month of, my notes say June, but I'm looking at a date that says May 19th. So I'm Nate Huffsney, currently a PhD student at Utah State. I will be moderating today's webinar with Sean Charles. I hope some of you had a chance to attend yesterday's webinar celebrating American Wetlands Month. The webinar was a joint venture with the Association of State Wetland Managers, the U.S. EPA, and the National Oceanic and Atmospheric Administration, as well as U.S. Fish and Wildlife. So please look into the Associated State Wetland Managers or the SWS website for more information if you were not able to attend, try to find that recording. So today, the general format we're going to take is going to be about a 40, maybe 45-minute presentation from our speaker, Sean, with about 15 minutes of Q&A from all of you. And be aware that we will announce the next webinar at the end of this session, so if you're not looped into the scheduling, we will drop that next scheduled event for you. And before trying to advance the slide. Let's go back a couple. We'll go forward one. A couple housekeeping items. For those of you who don't use GoToMeeting regularly, there is an attendee control panel where you can control the audio for the webinar. You can use this through your computer or you can call in on the phone as well. Please just consult the directions in the audio pane if you're having trouble so we can get through this smoothly. At any time during Sean's presentation, feel free to type in a question that you want to ask the speaker in the questions pane. As the moderator, I will queue up the Q&A and we'll read those to the presenter so everybody can hear the question as well as the answer. I should also note that all attendee phone lines are on mute to cut out background noise and distractions. And this webinar is being recorded. You'll receive a link to the recording following the webinar, and you can follow this up at sws.org. We also ask that you take a moment to complete the evaluation survey. As you may be aware, this is a relatively new effort from the Society, and this will just help us plan future events, have compelling speakers that meet the membership's diverse and varied interests. So on that note, I would like to introduce to you today's presenter, Sean Charles, PhD candidate, Florida International University, where he's working with Dr. John Kamenowski. You can find, as you see, a link to his work and Dr. Kamenowski et al.'s work at kamenowskilab.wordpress.com. And before we go too much further, I should let you know that although Sean is now a Florida, Everglades, and Texas wetland individual, he's originally from Williamsburg, Virginia, and he, let me cue this a little bit better. Originally from Williamsburg, Virginia, he followed up a career, got started early in environmental journalism, realized that he'd be a much stronger journalist if he had a strong scientific background. So he came to investigate the ways that biogeochemical changes are associated with sea level rise and vegetation shifts caused by global climate change, and how those alter carbon storage and vulnerability in coastal wetlands in kind of a non-traditional way, which I think is cool because Sean has very diverse skill set, which he will show you today. He's a very active member in the Society. He's currently the student rep for the Education and Outreach and Wetland Ambassador Committees. And before he got involved in that, he wrapped up a master's degree in 2013 with Jim Perry at VIMS, the Virginia Institute of Marine Sciences, where he looked at the development of functions in created forested wetlands and analyzed their success in the compensatory mitigation process. So if you can't tell, Sean is a guy who, if he had an airplane, he would be dropping logic bombs just about all day. So the work he's going to present to us, some of those logic bombs, will come from work that he did with Dr. Steve Pennings at the University of Houston, co-PI Dr. Ann Armitage at Texas A&M Galveston, and of course John Kamenowski. So he is presenting on behalf of a large and diverse team. And on that note, I will click over and I will say, take it away, Sean.
Okay. Hi, everyone. So thanks a lot for the Society of Wetland Scientists for inviting me, and for all of you for listening. At least I think there are some of you out there listening. I will say that sitting in a room by myself speaking to my computer sure makes telling jokes a little awkward, but it's certainly not out of the ordinary for my jokes to be greeted with silence, so I think I'll be all right. So my name's Sean Charles. I'm but a lowly PhD candidate. It's amazing to me that I'm included in this series with the impressive list of speakers that have given webinars thus far. To the extent that, to be honest, when I was first approached about giving a webinar, I thought the committee meant they wanted me to recruit someone. So I agreed, only to realize later that I was actually giving the talk. But I'm really happy and honored to be here. I'd like to say once again, thanks for the opportunity. All right, so my name is on this title slide, but really this research was done by a host of people from a number of universities. Particularly, I'd like to thank my advisor, John Kamenowski, and Steve Pennings and Anna Armitage for bringing John and I into this project, as well as the rest of these amazing people that you'll see up on the screen, particularly any of you that happen to be listening at the moment. So especially thank you, Carolyn. So this is the beginning of the outline of the talk. First, I'll be introducing some of the vegetation transitions that are occurring both in upland and in wetland habitats across. Next, I'll briefly introduce some of the structural and functional changes associated with those vegetation shifts, particularly from the literature from uplands in this case. Then for the bulk of the talk, I'll introduce you to a study underway in Port Aransas, Texas, aimed at understanding how vegetation identity and cover alter coastal wetland structure and function, particularly focusing on microclimate, habitat structure and food webs, and carbon storage and accretion. So coastal wetlands provide a number of important services, including habitat and food web support that sustain commercially important and threatened and endangered species. On the right, there's a picture showing that they filter water so well, in fact, that they're often used as wastewater treatment facilities. So this is from a past SWS meeting that was in Orlando. So I'll acknowledge that this is not quite a coastal wetland, but the concept still applies. They're so good at buffering the coast that engineers have begun to incorporate wetlands into levee building to reduce storm surge and therefore the necessity to build levees quite as high. And most important in this talk, really, they're also really important in terms of blue carbon storage, often representing 50 times more carbon storage per area than upland environments. And in fact, carbon storage is not only important due to its potential to mitigate climate change, but also it's essential for coastal wetland survival in the face of sea level rise. Thus, the storage of carbon is supporting all of these other services that coastal wetlands provide as well. Thought we were going to advance. Okay. Major shifts in vegetation communities are occurring worldwide across ecoregions. So this map highlights grassland ecosystems in the colored areas and identifies areas where shrub encroachment is occurring with the purple dots that you see there. It's obvious from this map that this is truly a global phenomenon that's happening across ecoregions and appears to be something very important that we should understand the impacts of. In particular, many coastal wetlands exist in vegetation transition zones. Here the blue areas are aridity gradients in which vegetated coastal wetlands grade into unvegetated ecosystems due to changes in rainfall. Additionally, and maybe more pertinent for this talk, the red circles are areas in which winter air temperature creates an ecotone where tropical mangroves, which in general are very susceptible to freezes, transition into short-statured marsh vegetation. And then the purple areas combine both aridity and temperature gradients. To me, this map just is a great illustration that a huge number of wetlands around the world are potentially going to be subjected to vegetation change in the near future as climates continue to change. In zooming in a bit more in the Gulf of Mexico, just slight differences in minimum temperatures of only a few degrees basically create the difference between coastal wetlands being dominated by marshes versus mangroves. And if we zoom into the area of interest for this talk, the Gulf of Mexico, as temperatures increase, mangroves are likely to increase their range and may ultimately take over almost all of the Gulf of Mexico. So the square that I have there is just showing basically the only remaining area that with a 4 to 6 degree temperature increase would remain marsh. And zooming in a bit more, getting us familiar with the Texas coast that many of you will hopefully be experiencing in a couple weeks' time. This is showing Landsat imagery from 1990 to 2010 and shows that mangroves have expanded by 74% while marshes have decreased by 24%. Notice that this is not a zero-sum scenario, and the majority of lost marshes were not replaced by mangroves. So in general, we have a trend of mangroves increasing their range and taking over areas where they didn't previously exist. But it's also really important to note that mangroves also die back. So in northern Florida, which is kind of near the edge of their range, severe winter cold spells can severely reduce mangrove cover. In this study by Stevens et al., it shows that mangroves died back between 4 to 12%. An interesting concept there is that the deaths were not uniform. It had a lot to do with their local exposure. So those exposed to north winds tend to die back a lot more than those that weren't. And it's also important to note that freezing deaths don't just happen at the very extreme ends of mangrove range. So these pictures below here on the slide show mangrove die-offs based on freezes that happened in very extreme southern Florida. So Biscayne Bay and the Shark River Slough, both in very southern Florida, south of Miami. And you can see that in Biscayne Bay, we had this freeze that caused a major die-off in mangroves, but once again, it's not uniform. Some of those mangroves survived. So we're looking at a change in composition and a change in cover rather than a total dieback of mangroves and replacement from marsh. So with that in mind, it's really important to think about not just a total shift from one species to another, but intermediate phases along that trajectory as well. Furthermore, mangroves and marshes often integrate in dynamic ecotones driven by salinity, competition, natural disturbances driven by wind and waves, and also anthropogenic activities, as you can see down in the right. Anthropogenic activities are much more damaging in developing nations, but they're also a factor throughout the U.S. as well. And these major changes, as well as mangrove and marsh dynamics, make understanding the impacts of vegetation shifts and intermediate cover of each species important for managing coastal wetlands. Luckily, there have been some great studies that have begun to investigate the impacts of vegetation on ecosystem structure and function. The first impact I'll mention is changes to microclimate. So first off, trees create shade. I know, big excitement from that one, I'm sure. However, things aren't quite as straightforward, and vegetation structure and percentage cover are also very important. In sparsely populated shrublands, there are areas where bare ground exists, which then absorbs more heat during the day and radiates it back at night. Similarly, tree canopies can be insulating, thus reducing the likelihood of frost at night. These impacts can have very important environmental effects, and I'll talk more about those later. Woody shrub encroachment has also been shown to influence ecosystem structure. Often, shrub encroachment decreases vegetation richness, but also can drive changes in higher trophic levels, from ants all the way up to vertebrates, as this meta-analysis shows. The figure shows the log response ratio of woody encroachment on the y-axis and rainfall on the x. It's interesting and relevant for this talk to note that impacts are climate dependent. And vegetation shifts may also impact food web dynamics. In this case, our example is not directly looking at shrub encroachment, but shifts among woody riparian species. So as vegetation shifts occur between functionally species, here they're just represented as X and Y, the consumers that depend on them can shift as well, with implications throughout the ecosystem. So the impacts of vegetation shifts are dependent on species traits, which is important going forward. In general, woody encroachment leads to higher carbon storage in the soil as well as above ground. Often this is because woody species increase carbon storage through longer-lived biomass and the production of recalcitrant organic matter that persists in the system. However, again, it's important to note that this impact is climate dependent. As mentioned previously, coastal wetlands are impressive carbon sinks, storing more carbon than any other ecosystem, and the majority per area, and the majority of that carbon is stored in the soil due to high productivity and slow breakdown in anaerobic conditions, as well as often being is the fact that coastal wetlands are often depositional environments that receive organic matter from elsewhere. The other really important thing about storing carbon in the soil is that organic matter usually represents a large portion of soil volume. Thus, net gains in organic carbon are essential in soil accretion and therefore the ability of wetlands to keep pace with sea level rise. So in this Alongi figure, mangroves show higher average carbon stocks than marshes. But another study by Gail Chimura, who presented last month, showed that there isn't much of a difference in terms of accretion, and if anything, salt marshes edge out mangroves. So there's still uncertainty about whether a change from salt marsh to mangroves or vice versa is likely to increase or decrease carbon storage in coastal wetlands. As sea level rises, wetlands must increase their elevation or be lost to the sea. There's an optimal level of inundation for accretion rates, and when wetlands are submerged, suspended sediment is deposited at higher rates, so only underwater can they have deposition from the water column. And to a certain degree, plant productivity is stimulated in higher water level situations. However, when you get past a certain level of inundation, plants become stressed or even die, thus reducing the organic inputs to the ecosystem. Since organic accumulation makes up a large portion of overall accretion, if in areas with when you lose that organic matter input, you can have a situation where sea level rise can ultimately lead to transitions to subtidal environments, as you see here in the figure on the right. A large driver of that ability to accrete, though, is also in the sediment. So sediment contributes directly to accretion, and when it is deposited on the top of on the surface of the soil, it creates a cap on the organic matter below it that reduces the oxygen available and therefore the breakdown of that organic matter. So it's important to note for our study that the Texas coast is relatively low in suspended sediment. And another driver of accretion is tidal range. So with wetlands in lower tidal ranges, they tend to be more at risk from sea level rise. Since the Texas Gulf Coast is microtidal, has relatively low sediment availability, and has a relatively high rate of relative sea level rise, the USGS ranks it as very high in the Coastal Vulnerability Index. So make sure you get to Corpus Christi this year. Just kidding about this year. So how do wetland soils accrete? So both from above and from below. Surface elevation is often correlates with elevation gain, though not always. And roots can increase soil elevation from below, and have been shown to be the most important driver of accretion in a number of coastal wetland systems. Due to their recalcitrance, their placement in anaerobic conditions, and their ability to bind the soil. And plants also influence carbon storage, so this is really important when we're thinking about changes in vegetation communities in coastal wetlands. Plants may differ in productivity and thus the quantity of organic matter produced. They may produce physically and chemically distinct organic matter that breaks down and is respired to the atmosphere at different rates. And they might allocate biomass differently. And this is really important for carbon storage because different allocations of their energy lead to different breakdown rates. For example, wood and roots break down much more slowly than leaves. So a plant that puts more of its energy into its roots rather than its leaves may have a higher contribution to carbon storage. They also influence the deposition of organic matter from outside, so allochthonous organic matter, by trapping material at different rates. And they can alter the microclimate, and thus altering breakdown rates as well, which I'll talk about again a little later. Finally, they prime and aerate soils to different degrees, and relevant for us, mangroves are known to aerate soils. This figure from Karen McKee shows that pneumatophore density is negatively correlated with sulfide concentration. So basically what we have here is that in areas with high numbers of pneumatophores from mangroves, you have more oxidized soil, which can therefore lead to faster breakdown of organic matter in that soil. However, it's unclear how mangroves will likely influence soil carbon storage. So this figure from Eric Yondo et al. Shows that there is no change there's no difference between soil carbon storage between mangrove and marsh communities in either North Florida or Louisiana. But in Texas, at least with taller trees, there's an increase in carbon storage in the soil compared to salt marshes. So we'll return to this again later as well. So now I'm going to switch over to talking about some of our recent research. This is an ongoing project in Port Aransas, Texas, and I'll be reporting some of our findings from a paper currently in review in ecology. It's co-authored by all of these lovely folks, led by the lead author, Dr. Hung-Yu Gu. Ultimately, our research is asking this broad question. How does mangrove cover alter ecosystem structure and function? And I'll note that while these pictures show a full shift from marsh to mangrove, we're really studying the intermediate levels of mangrove cover as well. So we set out to create a large-scale experiment in Port Aransas, Texas. It's very close to Corpus Christi, just about 30-minute drive away. It's a lovely place to visit, go to the beach, kayak, or take part in a dolphin tour that is attacked by pirates every day, and every day they're repelled by children with water guns. So it's quite an exciting thing to happen just outside of your research plots. A daily source of excitement, especially when you get caught off guard by cannon fire and think that it's perhaps an exploding vessel carrying oil. So, getting back to it, a lot of what we know about changing ecosystem function associated with different vegetation types has been developed by comparing existing ecosystems. And while this is hugely useful, these studies sometimes run the risk of confounding the effects of the vegetation with the underlying reason vegetation exists there in the first place. So for example, by comparing a natural marsh with an adjacent natural mangrove, the vegetation may be the cause or the effect of the difference in function. So i.e., maybe mangroves are more likely to establish in areas with higher soil carbon in the first place. So to avoid that risk, we went to the Aransas National Estuarine Research Reserve, established 10 24 by 42 meter plots, which at the time were all mangrove. We manipulated the vegetation, I'll explain that, to represent plots that range from 0 to 100% mangrove cover on 11% increments. These pictures on the right show are aerial photographs that show the treatments after they were immediately established. So I'll talk about the specifics next. So how does it work? Well, we identified the plots. Note these are all mangroves at the time. Then we created a grid system that created distinct 3 by 3 meter patches that are here outlined with flagging tape. And then in individual patches, mangroves were either allowed to exist or they were cut down. And I know what you're probably thinking. I can't believe you were allowed to cut down mangroves. And I understand that too, living in Miami right now. But there's a simple answer of why we were allowed to get away with that. And that's just that it's in sweet, sweet Texas. And I do love mangroves, and I feel bad to hurt them. But in my defense, I wasn't even there to do it. So no, instead, after a lot of work from some hardworking Texans and honorary Texans, I flew in from Miami, and in typical Miami fashion, decided to start talking on my phone in the plot. No, I'm just kidding. I just wanted to use this picture to show that after a couple of years, the cleared areas have largely been colonized by marsh plants and are no longer bare compared to the aerial photos from the last slide. Here's another way of visualizing the plots. So you can see the 3 meter by 3 meter cells, and basically they were systematically cleared, and this one sums up to 66% mangrove cover, but they were all basically gridded in this same way, and then mangroves were cleared to set up plots that had varying levels of mangrove cover. So now to some methods. First, we're measuring microclimate at weather stations in each plot. So just so you know how we've laid out these plots, down on the bottom right is an image of what it looked like right after it was cleared. Once again, no longer looks quite like that. And the black square in the middle is approximately where the weather station would go in each plot. You can see an example of that on the top right, where we measured air temperature, soil temperature, wind speed, and wind direction. The blue lines in that bottom right figure are the transects that we set up. And the first thing we measured there was light interception. The first thing I'll talk to you about is light interception, which basically every meter along those two transects we measured the amount of light reaching the soil surface, and then the amount of light reaching above the canopy, and determined what the level of light interception was. And in those same transects, we measured plant cover and rack distribution. Rack being particularly macroalgae and seagrasses. And then finally, before anyone went to the plots each morning, we would record birds within each plot. And the data I'll show are from 33 days of observation over the course of a year. So to some results. I'll show a few graphs like this during this section of the talk. What you see is percent mangrove cover on the x-axis and our targeted response on the y-axis. So to start with, in the top left, wind speed was highest in plots with low mangrove cover and decreased rapidly, particularly to 33% mangrove cover, and then declined more gradually after that. So, in essence, what we have is that mangroves blocked the wind, and they did so even at quite low densities, and we'll talk about that again in a minute. Light interception, just below that on the bottom left, shows a linear trend of increasing as mangrove cover increases. So that's light interception again. And then interestingly, the combination of these two factors leads to a nonlinear pattern of both average air and soil temperature, in which temperature peaks at intermediate densities. So why do we care? Why is this important for the ecosystem? Well, the first reason is that organic matter breakdown increases under warmer conditions and is also stimulated with light intensity. Thus, changing microclimate may influence carbon storage. I'll talk more about that later. Second, tall mangroves can outcompete short-statured plants and reduce benthic algae through shading. Third, mangrove forest development favors mangrove survival, potentially leading towards hysteresis. What I mean by this is, as mangroves increase their cover as well as their size, they influence the microclimate to become more favorable for mangrove survival. And that's the previous paradigm, but what we're seeing in this study is that the change in temperature is actually nonlinear and peaks at intermediate cover. And then finally, due to interaction with winds and waves, mangroves may intercept nutrients, organic matter, and seeds, causing a phenomenon that's been shown to create resource islands in upland shrub environments. Next, we found that marsh plants rapidly colonized the open spaces, but that patterns were nonlinear. So the first graph, the top graph, we saw that marsh vegetation cover decreased rapidly from 80% mangrove cover, from 80% at 0% mangrove cover down to 20% at 50% mangrove cover, and then became more gradual. We also found that the Bray-Curtis dissimilarity, which is a measure of beta diversity, increased with low-level mangrove cover and then decreased as mangroves became more dominant. In the bottom slide, we additionally found that rack distribution, in terms of how far from the water's edge coastal organic matter reached, decreased rapidly up to 45% mangrove cover and then declined more gradually. So this is, we did these surveys along the transects and looked at how far rack was distributed into the plots, found that it decreased with more mangrove cover. And then finally, we found that bird observations decreased as mangrove cover got higher, but once again, the change was nonlinear. And the majority of these birds were wading birds from the heron, rail, ibis, and sandpiper families. So what does this mean? Well, mangroves compete with marsh plants in a nonlinear way, reducing their establishment severely with low levels of mangroves. This phenomenon may be due to the aforementioned resource island effect, or mangrove shading adjacent areas, or finally, due to high levels of competition below ground in which mangrove roots extend well beyond their canopies. Beta diversity increases at first and then drops quite rapidly. So in an encroachment scenario, initial increases in beta diversity may be reversed with greater mangrove cover. And then there's a reduction in rack availability as mangroves become more dominant that reduces the inputs, may reduce the input of coastal carbon, which we'll return to, likely reduces nutrient subsidies as labile rack breaks down and likely influences, and finally likely influence consumer dynamics as rack becomes less available as a food source. In terms of bird use, different birds use marshes for different purposes, including foraging versus shelter. So most of our observations were of foraging wading birds, which seem to prefer the open marsh, but we have also seen rails nesting in mangrove trees. Thus maybe there's a trade-off between providing foraging versus nesting areas. Similarly, changing vegetation will affect bird species in different ways. And one really important local species that you almost have to talk about if you're talking about this area is the federally endangered whooping crane, which is the tallest bird in North America, and it stands at almost 5 feet tall. So it's an amazing, amazing bird. And the last migratory population overwinters in the Aransas National Wildlife Refuge. So any change in that species may be deemed much more important than more common species. Finally, we may have a change in food webs, so particularly right now thinking about the things that these wading birds might be eating. So for an example, this is just preliminary data that shows these are not our research areas, but they're nearby marshes. And you can see on the left you have an example of pure marsh stands, so no mangroves, and then an area with both mangrove and marsh. And this is from doing seine netting adjacent to these marshes. And the important things to notice are that the abundance and richness are similar, but the composition is different, with marshes having more gammarids, mostly amphipods, and the mixed mangrove marsh area having more gastropods and bivalves. So we have a change in the nekton community, which may have important impacts that are important in and of themselves and may have distinct implications for higher trophic levels that appear to the public more so than perhaps gammarids, which I had to look up. So this section I'll spend a bit more time on talking about carbon storage, because this is really where I focus my work in the research area. And this is also data that's mostly not in the previously mentioned paper. So for these methods, I set up 8 sampling areas in each plot. So remember, there are 10 plots, and they're represented by the 8 red squares at the bottom in the bottom image. They represent both mangrove and marsh-dominated patches, and they're set up so that they get both the areas close to the coast and farther back away from the coast. And for these, I'll be mostly comparing mangrove to marsh areas, but they also do often scale up to the way that we were displaying them earlier. I'll show you that in a second. So first, I looked at total organic carbon stocks. So this is above ground biomass, below ground biomass, surface soil carbon. Then I looked at the rates of inputs of carbon, and this was root productivity and surface accretion above feldspar plots. And then I looked at the rates of loss of carbon. So carbon breakdown of native litter, so in our cases it's black mangrove or Avicennia, and then the succulent Batis marittima, which was our dominant species in the marsh areas. I also looked at the breakdown of native roots, and then to normalize for species-specific differences in physical and chemical plant properties, I used wood and cellulose standard substrates, both above and below ground. So for most of these results, I'm going to use this background to help everyone stay oriented. So on the left, we have our mangrove areas. On the right, we have our marsh areas. And you'll notice that this is kind of an interesting and peculiar area of the coast where we're in a semi-arid place, and so the dominant species are mostly succulents, leading with Batis marittima. And the first result is that there was an order of magnitude more above ground biomass in mangrove patches, which wasn't a huge surprise based on just visually inspecting the sites. And that soil organic matter was also higher in the top 10 centimeters of the soil. Next, below ground, in below ground biomass, mangroves had about twice as much root biomass as marsh areas, but this was totally driven by the deeper area of the soil, the deeper area being 15 to 30 centimeters. So marshes and mangroves were similar at the surface, but then at 15 to 30 centimeters, mangroves had higher biomass, which is important both for carbon storage and also because having deeper root systems can enable you to survive better during drought periods potentially. Next, I looked at the inputs of root biomass and found that there was actually no difference between the mangrove and the marsh, but that mangrove roots were longer lived, which means that over time they could potentially lead to the accumulation of more root material below ground, living root material at least. The current paradigm in coastal wetland accretion is that higher biomass leads to greater accretion, as there's both more autochthonous plant material created by that higher biomass community, and because biomass interacts with suspended solids in the water column, causing them to be deposited at higher rates. However, in our sites, even with 10 times more biomass in mangrove areas, we found no difference in accretion comparing mangroves to marsh. Interestingly, though, we found that as mangrove cover increased, so this is back to the original figure format from before where you have mangrove density on the x-axis and then the response in the y-axis, and I found that in terms of both the depth of surface accretion and the amount of organic carbon accumulating on the soil surface, as you got more mangroves, you got a decrease in the amount. And we attribute this as well as the decrease in rack depositions in the marsh interior to dense mangroves limiting coastal subsidies by intercepting them at the marsh coastal edge. So this may largely cut off allochthonous carbon inputs. Of course, as we mentioned before, it's important to think about the environmental situation that you're in and ask, so would this be the case if systems weren't microtidal? And the answer is probably not. Higher water levels would likely allow more subsidies to pass above the friction, causing pneumatophores and better penetrate the marsh. So next I wanted to show this picture for a couple of reasons. The first is that I think it's strangely beautiful. Second is to point out that the wetlands often accumulate trash. Sometimes we've joked that trash should be a wetland indicator. But this area of Texas appears to have a real trash problem, which to its credit they're attempting to address, but with an aggressive public awareness campaign. But a lot of the wetlands in this area are pretty trashy. Finally, it's useful as evidence that accretion is occurring due to subsidies from the coast. So this wetland continues for a few miles towards the interior, away from the coast. And unless the lighthouse keeper is really messy, the only logical option is that this trash came from the coast. And sure enough, once again we can see that as you get higher mangrove cover, the majority of the rack is caught in the first 4 meters from the coast. And this is an important point. So if most sediment was coming from the interior, then the mangroves along the coast may create a barrier that keeps sediment in, thus increasing accretion. But in our case, it appears to create a barrier that keeps sediment and coastal organic subsidies out. So there may be major differences between the impact of mangrove cover between different coastal wetlands. Next, I used standard substrates, in this case cellulose, to look at the breakdown in patches dominated by mangroves versus marshes, and found that those in marshes broke down more rapidly. This is probably because we had underneath those canopies of mangroves, we had slightly reduced soil temperatures, and because temperature is a big driver of breakdown rates, that may have decreased the breakdown of carbon in mangrove patches. We also found that mangrove litter is way more recalcitrant. So after about 3.5 months, almost all of the marsh material, the Batis meridima litter, is gone, whereas mangrove litter is still about 80% of the mass remaining. So basically this leads to mangroves reducing the breakdown of organic matter on the surface by two different ways. Firstly, through decreasing the temperature, and second, through creating more recalcitrant material. And this has been seen elsewhere too, where there are differences between different species types in terms of their breakdown. And then those species types, when moved to warmer or cooler areas, also show a difference in their breakdown rate. It's just interesting in this scenario particularly because the mangroves themselves are the ones influencing that breakdown rate. Similarly, mangrove roots broke down more slowly than Batis marittima roots, also probably due to differing chemical and physical properties. Interestingly though, when I used wood standard substrates below ground, they actually broke down more quickly in mangrove areas, so indicating that while mangrove produced organic matter is likely to be recalcitrant, mangrove roots themselves alter the soil environment in a way that increases organic matter breakdown. So probably what I was mentioning before with their ability to aerate soils, that drives an increase in breakdown rates. This may create a trade-off in which mangrove roots cause the oxidation of existing organic matter while simultaneously adding new recalcitrant roots to the soil. And the overall impact of mangrove encroachment in that kind of scenario may therefore be influenced by the existing edaphic conditions in the marsh itself. So in our case, soil organic matter is really low in this area to begin with. So the input of carbon from roots may be more important than the increased rate of breakdown in the surrounding soil. However, that may not always be the case. So what might this mean in terms of mangrove encroachment's impacts on carbon storage in the Gulf of Mexico? Well, two studies from right near our site have shown that mangrove areas may accrete and store carbon at nearly twice the rate of marshes. And this is likely due to slow organic matter breakdown, as increased storage reflected increased lignin content in the soil, which is very slow to break down, particularly in anaerobic wetland soils. So that's from the area closest to us. So it appears that in Port Aransas, we do see an increase in carbon storage with mangrove establishment, and our research tends to mostly reflect that as well. On the other hand, other studies from the Gulf of Mexico have not found a similar pattern of carbon storage with mangrove establishment. So this study is from Perry and Mendelson that compared photosynthetic rates of Abyssinian spartina in Louisiana. So once again, very close to coastal Texas, but different species and probably different results. So they found that individual leaves of spartina were more efficient at photosynthesizing, but the mangroves had more leaves overall, and thus the overall photosynthetic rates were about the same. And therefore probably so were the inputs of carbon to the soil, although there's other factors that we're not accounting for there. In another study from Henry and Twilley, they found differing effects of mangrove establishment at different sites. So they found that in their first site that the soil organic matter content increased in more recent times once mangroves have been established. But in another one, they found the opposite effect. So these impacts must be largely influenced by location of site, as well as just the idea that mangroves are encroaching. And I think one of the major takeaways from all of this is that a lot of the effect of vegetation regime shift is dependent on the interplay between environmental conditions, one important one being climate, the species traits of both the species being replaced and the ones doing the replacing, and on the edaphic conditions of the site. So for example, in Port Aransas, we have a semi-arid microtidal marsh where a much higher biomass and more deeply rooted mangrove species is taking over from a low biomass succulent marsh, and you can see that in this figure from Yondo et al. That shows way less biomass in central Texas than in either of the other two sites or areas that he studied in North Florida and Louisiana. Furthermore, it's in a very low organic matter sandy soil. So can the impacts of this mangrove expansion be extrapolated to the rest of the Gulf of Mexico? Not easily. The results are likely to have a high level of ecosystem specificity, which is great because, well, job security. But I don't think the idea that results are case-specific is a cop-out. I just think that it means we have to match changes in important carbon storage processes to the specific scenarios of the area in which vegetation is shifting. And a really cool place to start is to look at how specific wetlands accrete. So this graph from Karen McKee shows that among seven mangrove types, elevation gains and losses were distinct from one another. Some had more mineral inputs, some more root, some more surface organic accretion, and drastically different levels of compaction, part of which is decomposition. So by understanding the importance of different mechanisms of carbon storage and processing and the likely impacts of these processes under different scenarios, we'll be better capable of predicting the impacts of vegetation change on carbon storage and accretion. So overall, I'd like to conclude on what we found in this study. The first is that we found that mangrove removal had strong effects on ecosystem structure and function in only 2 years. We also found that many of those changes were nonlinear and have determined based on logic bombs, thank you Nate, that these are often probably case dependent. We found that mangrove cover influences the microclimate by reducing wind speed, intercepting photosynthetically active radiation, and altering temperature in nonlinear ways. We found that mangrove can influence habitat, both through the structure and vegetation dynamics in terms of competition and establishment of marsh plants, through changing food webs and through bird use. And we also found that mangroves can influence accretion and carbon storage through having larger carbon stores, restricting coastal subsidies, producing recalcitrant organic matter, and reducing surface breakdown, but also they increase below ground breakdown. So the overall impact of that may end up being case specific due to that fact. So with that, I would like to thank everybody that helped, particularly the PIs from this project, John Komanowski, Steve Pennings, and Anna Armitage, as well as many others, and the universities that made this possible, our project funding, as well as my personal funding sources. And I have a plug to make for SWS's new initiative called Wetland Treasures. This is a project that came out of the Education and Outreach Committee and is now kind of taken on a life of its own under Abby Turna. But basically, you should check out the website, wetlandtreasures.org, and nominate a wetland. The goal of the Wetland Treasures initiative is to promote awareness, appreciation, and knowledge of wetlands, and to point out some of their features and exemplary functions and services. And they came up with all these cool icons for which function and services each wetland provides. And so this is what the website looks like, and by following these steps, you can nominate a wetland yourself, which I'd love for all of you to do. So with that, I'd like to show you some cool pictures that are from the area and hopefully get people excited about going to this area of Texas. Say thank you to everybody that made this project a joy to work on, and thank you to SWS for having me today. And I'll take any questions.
So Sean, let me be the first to thank you for your talk today and congratulate you on a job well done. We've had a couple questions roll in. If you have some in mind, please do not hesitate to type those into the question bar. So John Marshall asked pretty early in the talk, Why do mangrove scrub shrub have a competitive edge over emergent marsh with increased temperature?
So the kind of the basic answer is that once they reach a certain height, then they have an advantage because of shading. Um, Marshes can often outcompete mangroves at the seedling stage, so by stopping them from becoming established. But also mangroves are a tropical species, and they tend to have higher efficiency with their photosynthesis at slightly higher temperatures. But the big thing is that once they get established, they can grow for long periods of time and eventually get tall enough to shade out marsh species, and the cold temperatures knock them back and kill them off.
Thanks. And Anthony Curtis asked us around 11:30, did you consider elevation change in your work? If so, were R sets installed at any of your sites?
So we have sets in the potential next round of funding, but we didn't have any for this. We did have kind of the poor man's version of sets, which are elevation pins. So this is basically using PVC to determine how erosion and increased elevation occur. I would have loved to have them, but we didn't, basically.
And is there any plan in the future to kind of do a little bit more fine-scale elevation, accretion, erosion sort of work?
Yeah, so like I said, we have sets in the funding for the next cycle of this, so hopefully that will work out.
So stay tuned, in short.
Stay tuned, yes.
And then John Marshall also asked us toward the end of the talk, and I meant to tag down which slide, but it was pretty late in the talk. He asked, ellipses, or might the transition marsh to shrub be better explained by fire suppression?
So it certainly is in certain areas. We don't really have any evidence of that occurring here where we're talking about, but there's evidence of that occurring in the Everglades, in Australia, in quite a few different areas. The how they actually affect that marsh to mangrove transition, though, is sometimes unclear. So we've seen examples of that, or what you would expect is that you get a fire, it kills off the mangrove trees, and therefore marsh becomes more established. But often we've seen the opposite occur as well, including in the Southwest Everglades. So more work needs to be done on that as well. There's a lot of other reasons too, in terms of their ability to deal with drought, the effects of carbon dioxide, particularly when you're talking about C4 grasses versus a C3 mangrove tree. So there are a lot of potential climate change drivers that are causing mangrove encroachment or change. But in this case, it seems to be that the dominant impact is based on a reduction in freezes.
Right on. So Robert Nabel asked, and this kind of builds off of your last answer, he asked, do you think there is some way that the Earth is working to regulate global warming by increasing carbon sinks? And I am guessing, Robert, if you could clarify by typing into the question box, you're asking are plants in some way acclimating or adapting over time to their new environmental conditions to then be more efficient at making a living in these environments, sequestering more carbon? Is that the gist of the question, which, as written, was, is there some way the Earth is working to regulate global warming by increasing carbon sinks?
I see what he's getting at. So I've thought about that a lot, and it's a beautiful idea that this is kind of a Gaia hypothesis thing where it's basically driving, it's trying to correct the issues involved by increasing carbon storage in areas where it couldn't happen before. But unfortunately, I don't think that mangroves encroaching into marshes has anywhere near the same impact as, say, permafrost melting in the Arctic or the release of methane hydrates from the deep sea or any of those sorts of scenarios. So maybe on a small scale, I like the idea, probably not.
Fair enough. And then if anybody has any more questions, I'm going to go ahead and ask them. We'll have a few more, well, I say we're at noon now, Mountain Time, which means that a lot of you are past lunch break, but I'm going to ask one question. If you've got a last minute one, please slide it in. But Sean, one of the interesting things in your work, aside from the findings, is in the kind of geopolitical landscape. We see a lot of people that have preferences for certain types of wetlands, certain habitat types for what they like to do recreationally. Is carbon getting a fair shake in the Texan political landscape? I mean, is this a consideration like it is in some of the more progressive states like Washington, Oregon, California, parts of the Northeast that are actively kind of trying to move the discussion forward, or is it still a wetlands or habitat first, carbon as a secondary or tertiary ecosystem service?
I love the question, and I definitely approach introducing our research to people differently in Texas than I do in some other places. So there's definitely way more interest in how our research is going to affect fishing. That's kind of the go-to for everybody. But if you can frame it in a way where you're talking about, and this is of course to lay people, not to people that actually are more involved in wetlands and science. But if you can phrase it in terms of the fact that having more carbon in the soil helps to increase the buffering from storms that a wetland provides, then there's a lot of acceptance and interest in it. So talking about global climate change can be tough, though a lot of coastal communities are certainly aware of the issue. But framing it in a way that people may appeal more to your audience, I think, is really important.
Right on. So no other questions have rolled in at this point. So in closing, I would like to thank you again, Sean, and I would like to encourage the 20-something of you that are still on the line to check out all the work that's come out of this project. It's a huge team. It's a huge effort. It's a lot of data. It's going to be a great paper. Check them out in Corpus Christi, and then kamenowskilab.wordpress.com. And you can also go ahead and just check out this webinar in some sort of recorded format later on. Pass it along to your peers, and let's hope that this discussion continues as we go to Texas for the SWS annual meeting.
Yeah, and I just realized that my email address is not on this slide, so if you'd like to email me, it's schar056@fiu.edu.
And now, I almost forgot, we were going to have the eminent scholar, Dr. William Mitch, give the June webinar. So we go from the next cohort of hot scientists to one of the classic guys who literally wrote the book. He's going to be talking about supporting the goals and objectives of the International Ramsar Convention on Wetlands in the U.S. I think some of these big international efforts don't get the attention in the U.S. they possibly could. So please tune in. Please check out Bill Mitch's talk on June 16th. It's going to be fantastic. And for those of you who are going to be heading to the SWS annual meeting, safe travels and have a wonderful, wonderful time.